Hi everyone, and welcome to your introduction to Bioinformatics and Evolutionary Biology. My name is Gillian Woolard, and I wrote and developed this content for this module with the help of Dr. Mike Wheeler. We are both course leaders for this module, so we hope that you enjoy it and come away with more knowledge about bioinformatic tools and how they can help us understand evolution than you started with. So let's begin. For centuries, scientists have been trying to understand how organisms are related and how they have evolved. Now, this is important for a variety of reasons, one of which is agricultural biology. This branch of biology is concerned with the science behind farming, and one of the biggest parts of farming is crop cultivation. One of the ways in which evolutionary biology can be important to crop science is by helping to solve one of the most pressing problems facing the 21st century farm, which is how do we feed a growing population in a rapidly changing world? So, for example, let us understand how a plant has evolved to withstand dry arid conditions. Now, this may help us in the future as climate change begins to cause higher global temperatures and decreased precipitation around the world. So as we understand crop evolution, we may be able to use that information to develop sustainable agricultural practices that meet the growing global demand for food. Another use of evolutionary biology is medical biology. Now, one of the biggest and most widely publicised problems this field is facing at the moment is the emergence of antibiotic resistance in an increasing number of bacterial strains. Evolutionary biology plays a very fundamental role in this field by aiding the understanding of how these genes have evolved in these bacterial strains and what we can do to prevent further incidences of antibiotic resistance. The final example I have for you is the use of evolutionary biology in conservation. Now, we will be going into this in more detail if you do choose to take the biodiversity module, but I will briefly introduce you to this topic as it's a very important one. Evolutionary biology plays a huge role in understanding biodiversity as it aids our understanding of how life on Earth has evolved and how that can help us understand over why 90% of life that has ever lived on this planet is now extinct. When we can take a look into the past and understand how other, perhaps more similar organisms have evolved and responded to climate change, we can predict how the current species that inhabit the planet will respond to the current state of climate change. Evolutionary biology can help us understand exactly what is a species, which is a surprisingly complex question, but more on this later. For now, we need to understand how we classify organisms and their relationship to one another. One of the oldest sciences that is developed to understanding the relationships between species and their evolutionary history is taxonomy. Taxonomy is the science of classifying living organisms into groups, and is based on the binomial system that was first proposed by Carl Linnaeus in 1735. Binomial in biology means two-part name. For example, Homo sapiens, Homo neanderthalis, Pandroglydites. I'm sure you can think of lots of other examples. In taxonomy, types of organisms, also called taxa, are grouped according to their shared characteristics or traits, and they are classified and ordered into 8 descending categories. Each category includes less organisms than the one before it. So on the left-hand side of the presentation, you can see the 8 groups that the organisms are placed into. Now, the groups are said to descend as you work your way down them, as each category holds less and less organisms, but the number of groups within each category increases. So we will work our way down using an example, and I think we'll use humans as a great example just to teach you how these classification systems actually work. So we'll start with domains, which is the biggest one. Now, it is accepted by most biologists that there are only 3 domains of life. They are archaea, bacteria, and eukaryota. So every organism on this planet fits into these 3 groups. So what differentiates them? Archaea and bacteria both contain only prokaryotic single-celled organisms, and they have no nucleus. To differentiate archaea and bacteria, taxonomists often have to turn to genetics and use a special kind of DNA called ribosomal RNA to differentiate them. But what separates bacteria and archaea from eukaryota is more clear, as eukaryota contains only organisms that have a nucleus and membrane-bound organelles. So hopefully by now you know which ones we fit in, and I hope you're thinking it's eukaryota. So we do. Humans fit into domain eukaryota. Next up is kingdoms. Now, depending on where you are in the world, you may have been taught that there are either 5 or 6 kingdoms. I'm in the UK, so I will be teaching you that there are 5 kingdoms, which you can see on the screen. The difference between the 2 systems is simply that the prokaryote kingdom is split into 2 bacteria and archaea, whereas everyone else keeps it as 1 kingdom. So moving on to our 5 kingdoms, the first 3 may be more familiar to you. Here we have our animals, our plants, and our fungi, represented by our panda, common spotted orchid, and toadstool. Now we've already mentioned the prokaryotic kingdom, which contains only prokaryotes, for example, bacteria, like our picture of a bacteria on the screen. The last group are protists. This group contains eukaryotic microorganisms that are often unicellular. You can have plant-like protists, animal-like protists, and fungi-like protists, but they are most importantly protists. Now we have a famous protist on the screen, and that's the amoeba. Another famous and far more dangerous protist is called Plasmodium, which is the protist behind the disease that is malaria. So, which group would you place humans into? I am really hoping you chose the Animalia kingdom, as this one was a little easier, but it's about to get trickier, so let's move on to the rest. Now, as you may have spotted, I've grouped the rest of the taxonomic groups into one page. This is because this is where it gets quite complicated. There is no definitive or widely accepted number of phyla, class, orders, families, genus, or species. This is because there are many different ways to classify organisms, such as on genetics, like we've already spoken about, using the ribosomal RNA, as well as on body plan or observable characteristics. And how you would classify these organisms does really affect where they are classified into. So there is no one way to do it. Now, let's, for example, take a look at numbers. So we started off with 3 domains and 5 kingdoms, and as we are working our way down the hierarchical groups, you can see that the numbers increase rapidly. Again, from 3 domains, 5 kingdoms, into 23,742 higher taxa. That will include domains and kingdoms, as well as phyla, class, orders, and families. We then go into 75,715 genus, approximately 345,053 species, and approximately 25,082 lower taxa. That's increased quite rapidly. Though that will include subspecies, which is a term you may not have heard before. Again, defining a species can be very difficult, and when they're very closely related and we're not sure whether they belong to the same species or whether they're different enough to be a subspecies, we sometimes use that term, and it depends on who you ask sometimes as to where they will be classified, because clearly taxonomy is a little bit of a more fluid and ever-changing science. But I would love to show you where we fit in, so we'll just move on to the next slide. And here we are. This is our taxonomy here. So I've put a chimp next to us as they are our taxonomic cousins, as in the organism that's the most closely related organism that's currently extant, which means currently still alive. And I've also included a pine tree just to show you how very, very different it looks from us. Now, we know we are not closely related to pine trees, but we are related, and that's important. So as you can see, all 3 of us share a domain, Eukaryota. Now, this is where we and the chimp and the pine tree, we kind of disappear from. They go on to become part of the Plantae kingdom, and we go on to become Animalia. So we'll now just stick with humans and chimps, and you can see that we belong to the same phylum. Both of us belong to Chordata. We belong to the same class. We're both mammals. We belong to the same order. We're both primates. We belong to the same family. We're both Hominidae. Now, this is where we differ all the way to the bottom, the genus. We differ at the genus level, where we are Homo genus, they are Pan. And then obviously we also differentiate at species, because once you've differentiated at the genus or somewhere higher up, you don't join up anywhere further down. So I thought I'd include the pine tree just to show you how different we are. Obviously nothing then matches up with us. We are very different. We just share a domain, and that's about as closely related to them as we get. So obviously if I was to include a gorilla, an orangutan, you would expect that again we share a lot more. If I start including a lemur or a fish or a shark, you would certainly expect that we differ higher up in the groups. So feel free to go on NCBI Taxonomy and take a look at the others and just search around and have a play. But for now, we'll just move on. So, now we've covered our own taxonomic record and we've had a whistle-stop tour of the various ways in which taxonomic rank can be decided, I want to delve into one of the most modern ways to classify organisms, which is to construct a phylogenetic tree. Now, phylogenetic trees are simply a way for evolutionary biologists to visualise relationships between taxa, which are the organisms at the end of the tree, and also their relationships between one another. Now, there is some debate as to who drew the first phylogenetic tree, and when, but it is widely thought to have been first drawn by Augustin Aguirre in 1801 when he drew the first one to show relationships between plants. He was then closely followed by Edward Hitchcock in 1840, Charles Darwin in 1859, and Ernest Haeckel in 1860. The pictures of all of them can be seen at the bottom. But obviously back in the days when these first phylogenetic trees were born, DNA technology didn't exist. So what did they use? So instead, many of the original phylogenetic trees used physiological traits such as the presence of wings, flippers, legs, whether the species had a similar structure in their anatomy, for example, maybe the eye. Can you see a problem with this? Now, all of these organisms have wings, but do you think they're closely related? I hope you said no, because although they may have wings, they are simply not closely related. Many organisms living in similar environments have evolved similar structures, for example, thick fur in cold environments. Like our example earlier, some have evolved wings for life in the air, others have evolved flippers for life underwater, and like us, legs for life on land. However, that does not mean they are closely related. Similar traits, structures, or behaviours can evolve independently many times in distantly related lineages. So, as we already mentioned, wings have evolved independently in birds, mammals, and insects. Similar environments instill similar selection pressures on organisms, which drives independent evolution of these traits, structures, or behaviours. So here is another great example of convergent evolution that I was completely fascinated with when I was an undergrad. This paper is from Yoshida and colleagues, and it shows that human and octopuses have both evolved the camera eye independently. So despite the fact that we are very distantly related and we differ at the phylum level, we both have the camera eye. So instead of using shared physiological traits and similar physiological structures that we used to draw the very first phylograms, we now use genetic sequence. Sequencing the genetic code of organisms has become less expensive, which means more organisms are having their entire genome sequenced, which means more DNA sequences to compare, which is all good for us. So we base phylogenies using the genetic code on the basis that organisms that are closely related will have more similar genetic sequence in common with each other than organisms that are distantly related to them. So we would expect a chimp to have more genetic sequence in common with us than a pine tree, or an octopus, for example. So we base phylogenies using genetic code on the basis that the ones that are more closely related will have similar genetic sequence with each other than organisms that are distantly related to them. So we would expect a chimp to have more genetic sequence in common with a pine tree, for example, or an octopus. So the more closely related to the organisms they are, the more genetic sequence in common. The more distantly related they are, the less genetic sequence in common. That's pretty much the basis. So what you can see here is 3 made up sequences that I've put together, and you can see that there are some differences between them that we've highlighted. So we have species 1, 2 and 3, and we want to know who is more related to who. Let's go ahead and take our genetic sequence and compare them to one another. So they're already aligned, and we can see that 1 and 3 have 25 out of 28 nucleotides in common, 1 and 2 have 21 out of 28 nucleotides in common, and 2 and 3 have 21 nucleotides in common. This tells us that 1 and 3 are more closely related to each other than 1 or 2 and 2 or 3. And this, ladies and gentlemen, is the basis for phylogenetics. It really is that simple. So now we know how to use our DNA to build a phylogenetic tree. What can a phylogenetic tree tell us about our own species? Over the next few slides, I'm going to cover a few key terms with you and teach you how to read a phylogenetic tree. This is a skill you'll be needing for practical later, where we will look at our own ancestry and look at other organisms that are closely related to us. So here you can see, this is just a very basic made up phylogenetic tree, and you might want to pause this video, print it out or make notes, but it's pretty self-explanatory. You can see that the species at the end are called taxa. We've already mentioned that. They're the present day species, they're extant. And we also have, in green, highlighted an example of a sister species or a sister taxa. They are 2 most closely related to each other than they are to the rest of the species in the tree. We have a root species, which is very, very important. When you build a phylogenetic tree, you always include an organism that's just slightly different from the rest of them, just to root your tree. It also acts as almost like a quality control, because if that organism groups itself in the tree, and you know they're not closely related, you know it's probably one of the programs has gone wrong somewhere, or one of the steps you've taken has gone wrong. So it helps to kind of root the tree, both for quality control purposes, and it does help form the internal nodes, which you can see in blue. They are our common ancestor and speciation events. So we often call the species or the end of the trees are external nodes, and the ones that are highlighted in blue are internal nodes. And at the bottom you can see time is kind of going towards the right, which is often in millions of years. You may see phylogenetic trees that are drawn in different ways. Don't panic, the information is exactly the same. You can see where they've put the tips, the internal nodes, and the root. It's telling you exactly the same thing, it's just drawn in a slightly different way. So outgroup species I've already discussed is a distant related to the other species in the tree. By having an outgroup, the phylogenetic software can reconstruct the evolutionary history of our species, forming our internal nodes. You may have come across other people using the term root species instead of outgroup. That's perfectly okay. Different people use 2 different names, but they mean exactly the same thing. And here we can see, we've already mentioned again, external and internal nodes. So external nodes tell you the present day extant species and their relationships to one another. Internal nodes tell you how the present day species got to where they are by showing us speciation events. Again, I've just quickly highlighted this one again because some people do find it quite confusing what the internal nodes actually stand for. They are our common ancestor, so species A and species B share a common ancestor, and species C and species D share a common ancestor, and they're also speciation events, which we will just go into a bit more detail. Now, speciation events are when one species splits into two or more. It is the driving force behind Earth's astonishing biodiversity, and it really is important. I want you to pause this video and watch another video made by Cornell University, and it explains speciation in such a great way that I think I could do it no more justice. So please do pause this video and go and watch the educational video. So hopefully you're back after watching the speciation video. We've just got to run through a few more key terms. So here we have what is referred to as a sister taxa. Sister taxa refer to the closest relative of a taxon in the phylogenetic tree. So in this example, 1 in 3 would be sister taxa. A clade is our next key term. A clade is a group that includes a common ancestor and all of their descendants. So we can see here that A and B and their common ancestor made a clade, but that B and C do not. So in a bit more detail, we can see that there are different kinds of clade. A monophyletic clade includes a common ancestor and all of their descendants. Our example here shows that A and B and C share a common ancestor and therefore make up a monophyletic clade. A paraphyletic clade includes an ancestor and some, but not all of their descendants, and we can see here that B and C make a paraphyletic clade. An example of this would be A and B and C share common ancestry, but A looks very different to B and C and their ancestor. And last but not least, we have a polyphyletic clade, which includes two descendants, but not their common ancestor. And we can see here that A and C make a polyphyletic clade. An example here would be that A and C have evolved a similar trait through convergent evolution, like perhaps wings, the eye structure. We've covered a few examples already. So I've just put my examples at the bottom if you do want to pause and just take another quick read through of these, because I understand that the terms at first are quite complicated. Here again, common ancestry. I told you it was important. In orange, we have the common ancestor of A and B, and in red, we have the common ancestor of A, B, and C. And here we have ancestral lineages. It may look complicated, but it really isn't. So let's go through it. So we have the tree's root at the bottom. It is the common ancestor of all of the species on this tree, which is A, B, and C in this example. Now, if we go in the direction, let's follow the lineage of species C, we'll see that this branch is the only ancestral lineage of species C, not A or B. If we go in the other direction from the root, we can see that this is the ancestral lineage of both A and B, and it led to their common ancestor. As soon as we branch off from this common ancestor, we can see the separate ancestral lineages for both species. So species A over here in purple, B in blue, and that covers ancestral lineages. So we've covered all our key terms, guys, and I'm just going to mention a couple of the things I think are quite worth noting. One of the problems with phylogenetics is extinct species. We cannot include most extinct species on phylogenetic trees. This is because fossils are rare, and getting enough good quality DNA from fossils is unlikely as DNA degrades over time. DNA has a half-life of roughly 500 years, and as most fossils are millions of years old, this would mean there is very little DNA left, let alone any good enough to use in a phylogenetic tree. However, some recently fossilised remains do contain DNA, and that we can use. An example of this is that DNA that's been extracted from a fellow human species, or subspecies, depending on who you ask, are the Neanderthals. So we can actually include these guys in a phylogenetic tree, although we won't be doing this in our example. So that's everything, guys. Well done. If there's anything that's unclear, please do go back through the presentation again, check the resource section, or do some independent research using a search engine, or if you're more of a visual learner like me, try a video site like YouTube. We've done the hardest bit, and after the quiz we can get to put all your newly acquired knowledge to the test by building a phylogenetic tree to look at the relationships between some species that belong in the family Hominoidea, the apes. So I'll see you in the practical, guys. Well done.